Great, Andrew. Thanks for that presentation. I especially like that you're focusing on new mechanisms of action. We heard this morning that that was a key. One of our speakers this morning mentioned how that's a key aspect of getting people excited about investing in molecules is when they have new mechanisms of action. So next speaker is Chi Lu, and she's been working to help the FDA understand how computational methods can improve early drug development. So this will be a good segue or good building on the presentation we just heard. So go ahead and take it away, Chi.
Thank you, Howie. Can you see my screen, please?
Yes, that's great.
Great. Good afternoon, everyone. It's a great pleasure to be here today and to share with you my thoughts on innovations in drug discovery and development for prevalent chronic diseases. First of all, I want to mention that I'm not a drug discovery expert. I'm a clinical pharmacologist and data scientist in training, and I currently work at the FDA. FDA does not play a major role in drug discovery. Instead, FDA primarily focuses on drug development and regulation, which logically follows the drug discovery phase. However, evaluating the recent products in drug development has given me a unique perspective about how innovations in drug discovery have led to novel treatment options and have improved public health. In addition, I have spent most of my time at FDA in the therapeutic area of oncology, which give me a front seat to witness amazing innovations that have transformed cancer therapy. So I'm going to talk about some oncology examples, and hopefully there can be some lessons we can learn and apply to other prevalent chronic diseases. Here's my disclaimer. Innovation is at the heart of drug discovery and development. Scanning the current scientific landscape, I think the current and upcoming innovations in drug discovery and development can be summarized into three major categories: new therapeutic modalities, new types of data, and new analytical tools. In today's presentation, I will talk about these three categories of innovation and share some examples. First, let us look at new therapeutic modalities. I will start with the targeted therapies. The figure here shows the number of FDA original approvals of new molecular entities from 2010 to the first half of 2019. The blue bars here represent the total number of new molecular entities, and the yellow line represents the number of targeted therapies, which are new molecular entities for which the initial approved indication is restricted to a subset of patients who are identified through a molecular testing. As you can see from this figure, the past decade was marked by significant growth in the development and use of targeted therapies. Biologics, especially monoclonal antibodies, are one of the fastest growing class of therapeutics. Here I listed immune checkpoint inhibitors as an example. This group of monoclonal antibodies has been transformative for the treatment of patients with cancer. The figure on the left shows the timeline of the FDA approvals of immune checkpoint inhibitors, including 7 drugs and 18 cancer types. As you can see, immune checkpoint inhibitors are becoming a more and more important part of cancer treatment. Dr. James Allison and Dr. Tasuku Honjo won the 2018 Nobel Prize for being the pioneers in this field. Antibody-drug conjugates and bispecific antibodies are also very interesting novel biologics. While in 2009, only one ADC drug was used clinically, in 2020, 9 FDA-approved ADCs are available and many others are in active clinical studies. In 2014, FDA approved blinatumomab for acute B-cell lymphoblastic leukemia. This marks the first U.S. approval for a bispecific antibody. This bispecific T-cell engager immunotherapy binds CD19 on the surface of B-cell lymphoblast and CD3 on the surface of T-cells, bringing the different cells together and driving an immune response to malignant cells. Currently, many other bispecific antibodies are under clinical development. Other new therapeutic modalities include cell-based therapy, oligonucleotide-based therapy, microbiome-based therapy, and viral therapy. Due to the limited time, I won't be able to go through all all of them in details, but I listed the FDA-approved cell-based therapies and viral therapies for cancer treatment on the slide in case you'd like to check them out later. It is important to emphasize that today's breakthroughs in oncology are the results of decades of investment in cancer research and drug development. The US government has made significant investment on cancer research. In 1971, President Nixon signed the National Cancer Act into law, which represented the US commitment to the war on cancer. In 2016, Congress passed the 21st Century Cures Act, authorizing $1.8 billion in funding the cancer moonshot over 7 years. The pharma industry also made vast investment. As shown by Susan in the first session, oncology is currently the largest segment of pharma industry. I think there might be several reasons for this. First, basic research in cancer has led to a number of promising targets and drug candidates. Second, there might also be a high financial incentive. These two reasons got confirmed in the first session. In addition, I think the flexibility and innovation in FDA's regulatory strategy for cancer drugs could also be an encouraging factor. Being at the FDA, I have witnessed how our oncology divisions have frequently used innovative regulatory strategies such as breakthrough designation, accelerated approval, and real-time oncology review to promote patients' early access to safe and effective anti-cancer therapies. I hope the entire society can evaluate what we can learn from oncology and apply them to other therapeutic areas. Now we have discussed new therapeutic modalities. Let us look at new types of data that can be used for drug discovery and development. Integrating multi-omic data can be powerful in the study of the complex relationship between the involved micromolecules and drugs. Multi-omic data broadly covers the data generated from genome, epigenome, transcriptome, proteome, metabolome, and microbiome. Another new type of omic data are radiomic data. Radiomics are emerging science that use algorithms to extract features from medical images, has the potential to improve disease detection, characterization, as well as assessment and prediction of treatment response. In the past decade, electronic health record system has been widely adopted, and smartphones and wearables make it possible for patients to generate a vast amount of health data on a continuous basis. We have an opportunity and the obligation to derive real-world evidence from real-world data to promote public health. FDA defines real-world data as data relating to patient health status and or the delivery of health care routinely collected from a variety of sources, including but not limited to electronic health records, claims and billing activities, product and disease registries, patient-generated data, and data gathered from other sources like mobile device. In 2018, FDA issued a framework to evaluate the use of real-world evidence in regulatory decision-making. There are also many novel experiment systems that can generate new types of data to facilitate drug discovery and development. Here listed an example, the microphysiological systems, which are a class of microfluidic platforms that recreate properties of tissue microenvironments. You probably have heard about the organ on the chip and tumor on a chip systems, which are examples of microphysiological systems. These novel systems can potentially be used for the prediction and assessment of drug PK, metabolism, safety, and efficacy. With the increased amounts and new types of data, new analytical tools are needed to transform big data into smart decisions. So in the next few minutes, I would like to briefly talk about a few new analytical tools that can play important roles in drug discovery and development. The first one I would like to talk about is Model-Informed Drug Development, or MIDD, which is referring to the application of exposure-based biological and/or statistical models derived from preclinical and clinical data sources to address drug development and/or regulatory issues. Many different types of modeling approach can be used for this purpose, such as physiologically based PK model, quantitative system pharmacology model, and quantitative structure-activity relationship models. Here I'd like to share an example where MIDD was applied for drug repurposing for COVID-19. The manuscript listed here were published by colleagues in our office. It is about how to connect hydroxychloroquine in vitro antiviral activity to in vivo drug concentration in order to predict the potential antiviral effect in humans. The conclusion from our analysis is that under a safe oral dosing regimen, it is unlikely that hydroxychloroquine can achieve high enough concentration in vivo to show antiviral effect in human. Our paper was published in May 2020. On June 15, FDA revoked the emergency use authorization for both hydroxychloroquine and chloroquine, and our analysis was listed as one of the reasons for this decision. As you have heard from Andrew's talk, AI and machine learning are powerful tools for drug discovery. They can be used to aggregate data, synthesize information, seek patterns, and optimize decisions. When used in drug discovery and development, AI and machine learning can help us understand the disease and targets, generate and evaluate drug candidates and combinations, improve clinical trial design, and advance precision medicine by improving diagnosis and treatment. Here are some examples of submission with machine learning components that FDA has received. In recent years, many applications for machine learning-based medical devices have been submitted to FDA. Most of them are imaging-based diagnostic software. This type of submission is reviewed and regulated by FDA's Center for Device and Radiological Health. At the Center for Drug Evaluation Research, we have received submissions with machine learning components in a number of areas. For example, we have seen submissions with machine learning-based disease models proposed as drug development tools. We have also seen submissions with machine learning to predict drug response based baseline factors. Some submissions try to identify predictive biomarkers for drug response. There were also submissions with machine learning applied to claims and EMR data to identify drug abuse-related problems in post-marketing setting. There's a famous quote by Dr. Andrew Ng, a leader in the AI field. It wasn't who has the best algorithm that wins. It was who has the most data for the application. To make the best use of novel analytical tools, we need data sharing and pre-competitive collaborations. In summary, we expect to see upcoming innovations in drug discovery and development in three major categories: new therapeutic modalities, new types of data, and new analytical tools. Investment from government and industry are needed for scientific breakthroughs. Data sharing and pre-competitive collaboration can potentially benefit everyone, especially the patients. In the pursuit of innovation, if we want to go far, we need to go together. Last but not least, I would like to thank my colleagues who have contributed to this presentation or the listed projects, and thank you for your attention.
Thank you, Chi. I really enjoyed how you talked about not only new tools that are being developed, but how they're actually being used in drug development and in the regulatory framework. So that was great.